Hello, students, and welcome to my screencast on DNA to help you prepare for your test. Just like all my other videos, this is under the Creative Commons license. It just means that it can't be shared for profit, but just to help you. It's meant to help you. All right, here you can see some pictures of the key players in the discovery of the structure and function of DNA. You see DNA being replicated, and right here I put a little blueprint, right? And it's just like it's the blueprint of life. All right, you might hear the singing in the background. I got some sick kiddos this weekend. I was really sad to miss the play because of that, but I will make sure to watch the DVD of it and get all the recaps from you in class. So let's jump right in. So we started out this unit talking about the great experiments to figure out the structure and function of DNA. We knew from Mendel and his work and Thomas Hunt Morgan that there were genes, and these genes can be passed down through generations. They don't blend, but you get different alleles from mom and dad, and you get a phenotype from that, right? Whether it's a purple or a white flower. For us, it could be, our blood types, etc. So first experiment we talked about was Frederick Griffith. Why don't you go ahead and pause the video and see what you remember about it? Could you sketch out the experimental setup? Like, what was the control group? What was the experimental group? What did we figure out from it? Could you ask questions about the nature of DNA? Okay, I hope you paused and tried that out. Right, here's a little background on it. He was working on pneumonia, trying to find a cure. He was mixing bacteria together, some of it that he'd killed and some of it that he hadn't, to see what would cause a disease in mice. He discovered that bacteria could pass something between each other, and he called that the transforming principle. So here we go. Here's what actually happened. The rough strain was non-virulent. It means it didn't hurt the mice. He put the rough strain into this mouse, and nothing happened. Mouse lived. The smooth strain was the bad guys. So he put the smooth strain into this mouse, and the mouse died. Then he heated up the smooth strain, put it into the mouse, and the mouse lived. So it means that he was able to kill the smooth strain and see the mouse live. Here is the transforming factor. Here he put the heat-killed smooth strain with the rough strain, and somehow the rough strain took up the gene or whatever it was that was letting the smooth strain kill the mice, and the rough strain became virulent and killed the mouse. Pretty amazing, right? I showed you a quick video in class that bacteria are able to get genes horizontally, right? We pass them down from parents to children, as you saw in my slide beforehand, but bacteria can pass them down the line and across. Pretty crazy. All right. So that's what Frederick Griffith found. Now we're going to get to the Avery experiment. Not many people did this poster in class, but it's a very important experiment. I'd love for you to pause the video and see if you can describe the experimental setup here and what Avery, McLeod, and McCarthy found. All right, did you get it? Here's what happened. Avery and company took these heat-killed S cells. Remember, if you put the heat-killed S cells into a mouse, it won't kill them, right? The heat killed it. Here they added an enzyme. You can tell it's an enzyme because it ends in ASE, and it's going to destroy all the proteins in the sample. So it destroys all the proteins. They then added the R cells, and the R cells became deadly. So this is a little tough for students, right? Because you have to think about negatives, and you have to think about like arrows and signs. It really helps to sketch it out, and it really helps to try and tell a friend about it. So if you destroy the proteins and the R cells become deadly still, it means that proteins were not the transforming factor. If the proteins were the transforming factor, then the R cells would not have become deadly. We didn't talk about this one too much in class, but they also tested for RNA, and so they destroyed all the RNA. Same thing as the proteins, the R cells became deadly. What that tells you, if you think back logically through the feedback loops, is that RNA is not the transforming factor. RNA isn't what's going from the heat-killed S to the R cells. Then he added DNase, deoxyribonuclease. It's going to chop up DNA. Right? This was interesting because you added the DNase, you added the R cells. The R cells were not deadly. So let's think about what that means. If you chop up all the DNA in the sample, it means that whatever was passed between the S cells and the R cells doesn't happen. The DNA isn't passed between them, and the R cells are not transformed. In other words, he's able to prove that transformation requires DNA. DNA is the genetic material of the cell. That's a tough one. You should go back through that one a couple of times and check it out. Okay. And then here's going to be the confirmation of the Avery experiment. It's the famous Hershey and Chase experiment. Here's the blender. Pretty cool little setup here. Just think about setting up an experiment like this, just using that scientific method. So fantastic. All right, these scientists, Hershey was a guy, Chase is a lady. They figured out that proteins will have sulfur in them. It's in methionine and cysteine, the amino acids. And phosphorus is only in DNA. So that's a pretty cool deductive reasoning, right? And then they're going to label those and see where it goes when a bacteriophage infects, which a bacteriophage is a virus when it infects bacteria. So before I go to the next slide where you can see the experiment in action, can you pause the video and try and remember and draw out the experimental setup? Draw what happened. Okay, hopefully you got something like this. In the first one, they labeled the radioactive sulfur. That's only in proteins. They labeled it in the bacteriophages. They let the bacteriophages infect the bacteria. They literally shook them off in a blender. Such a cool idea. And then they spun the materials, and they could see if it was in the bacteria, it'd be in the pellet at the bottom because that'd be more dense than the liquid. What they found is that the bacteriophage did not inject the radioactive sulfur into the bacteria, right? It stayed in the liquid. So it stayed out here in the viruses. In this one, they labeled the radioactive phosphorus in DNA. They let the bacteriophages infect the bacteria, these viruses. They then spun it. And this is so cool, right? They saw that the radioactive phosphorus was in the pellet at the bottom. So look at that. It's just a beautiful experiment, a beautiful setup. You can see that the transforming factor between these viruses and the bacteria had to be DNA. And the beauty of it is that they radioactively labeled this phosphorus and they could see it in action. We do this today. We'll put radioactive tracers in patients and see if it's concentrated in one area, if it's undergoing high metabolism, and that might mean a tumor or cancer. Okay, so those were 3 really big experiments. And then another thing that helped figure out the structure and function of DNA was Chargaff's rules. He figured out that A's are going to be equal to T's and G's are going to be equal to C's roughly in the proportion of DNA. And so this is pretty cool. Remember I told you ashtray, garbage can. That's what my 9th grade biology teacher told me. But smoking is not good. So you should think of a different way to remember that A's and T's match up and G's and C's. So it all came together, and then I gave you the talk about Rosalind Franklin and Watson and Crick and Maurice Wilkins and Gosling, and really the quest to find the structure and function of DNA. I'm not going to put that on this test, but remember what I taught you out of that, the whole point being that great scientists are not always great people, right? And credit is not always given where credit is due. And so I want you to not only be a great person, a great scientist, and just be a great human being. All right. So let's get a little bit into DNA. Just a quick reminder, right? We have these chromosomes. You're going to get 23 from mom, 23 from dad. What is a chromosome? What is it really? It's DNA with some proteins around it. So the DNA is wrapped around the proteins called histones. Right? right? It packages it nicely in a nucleus of the cell so that it's protected. DNA is made up of nucleotides, which are made up of a sugar, a phosphate group, and a nitrogenous base. You should know that the nitrogenous base order is what differs, and that's what gives you your characteristics and me my characteristics, right? Hopefully you remember the little TED talk that we watched that showed all the books that showed how much our human genome is composed of. It's just a staggering amount, right? If you're trying to look at just one base, this is how small it is. It'd be like if you're trying to pick up a nickel or a small coin if your hand was the size of the Middle East. It's a really small molecule and it packs a lot of information in there, right? And these sequences of letters equals your genes, which equals you. So you should feel special. All right. See if you can pause the video here and try and describe the structure and function of DNA and how it works in the cell. All right. We already went over that it's made up of repeating nucleotides, right? They're joined together by dehydration synthesis reactions. And it's kind of like salt and caboose highway. You have one strand going this way and another strand going the opposite direction. Once they figured it out, they were really able to crack the code. A's and T's and G's and C's always pair up together. You might have heard of purines and pyrimidines, and it's due to the hydrogen bonding between them. If you remember from our unit on biochemistry, hydrogen bonds are weak bonds, so we're able to break apart the DNA and read it and transcribe it and copy it, etc. And there's the famous double helix. Okay, we already went over hydrogen bonding. So now let's talk about DNA replication. Please pause the video, try and use a whiteboard, a piece of paper, etc. And write out how DNA is copied. Right? The real question, and if you got to it on any of the extension problems, is it copied dispersively? Is it copied conservatively? Is it copied semi-conservatively? And this was figured out what is often called the most beautiful experiment in biology by Messelson and Stahl. Okay, so let's go over and you can check yourself. Hopefully you got the DNA's unzipped by an enzyme called helicase. Helicase is going to break those hydrogen bonds and unwind it. An enzyme called DNA polymerase attaches to each strand. So if we break the DNA polymerase attaches to each strand and it's going to add on the correct nucleotides. It also proofreads as it does this. There's more enzymes than that. I had you model topoisomerase in class, which relieves tension on the winding coils, etc. And there's other ones as well. So as the DNA polymerase is adding, it's also checking it. You're going to get two identical copies of DNA. It's going to conserve. It's called semi-conservative replication. So here's the blue. That's the parent DNA. Let's pretend that we're making more skin cells, right? You shed skin cells every day. This strand, due to the ATGC base pairing rules, is going to be identical to the parent strand. We opened it up and we synthesized new nucleotides here. Nucleotides we get from the food we eat, and we can process and make them ready in our cells. An interesting tidbit, DNA polymerase can only add in the 3' direction. It's because that has an OH group, and so it's ready for dehydration synthesis when we lose a water molecule and join two molecules together. So this one is figured out by a Japanese scientist called Okazaki, has to go backwards, and it does loops. So that's pretty cool evolution piece right there. So if you want to learn more about that, I've got the DNA cartoon books you can check out in class, and of course you could sign up for AP Biology with me. All right. This is something I didn't emphasize well enough in class, but a little bit of the differences between eukaryotic and prokaryotic DNA replication. We have a lot of DNA in eukaryotes, organisms that have a nucleus. So therefore, we're going to replicate in both directions. Here, it's easier to see with a picture. So we're going to have multiple points of replication until the whole chromosome is copied, whereas prokaryotes are just going to start in a single point and continue in two directions until the entire chromosome is copied. I think it's really important to remember that both bacteria, eukaryotes, they both have DNA. DNA is foundational to everything that's alive. Your dog has DNA, trees have DNA. Sometimes I find students get confused about that. We'll kind of touch upon that in the universality of the genetic code in our evolution unit. So we just talked about how to copy DNA. Well, DNA stays in a nucleus. How do you get your looks? How do you get your complexion? How do you get your hair? How do you get the enzymes that are all over your body, etc.? Right? And that's going to be proteins. DNA is going to code for proteins. This is called the central dogma. And so I probably shared with you in class that this was my interview question for getting hired here at TASM. And it was to explain this and to go through this for a non-science person. And so I talked about DNA being a cookbook, and RNA being a recipe, and then the protein being the product. And then the amino acids are going to be the different ingredients that go into making that final finished cooking product. So this is called the central dogma of biology. DNA coding for RNA, which then codes for proteins. It's pretty cool. So let's jump in. Can you remember the differences between DNA and RNA? Go ahead and pause the video. I'm sorry, I should have animated this, but just cover it up with your hand and see if you can label them out. Okay, hopefully you got that RNA is single-stranded. It's got ribose instead of deoxyribose. And it uses U's instead of T's. Remember that RNA always, students, loves you. So what would be the RNA code to this strand of DNA? See if you can get it. Oops, I thought I had it. Well, it would be UAUCGC. All right. So we just made some messenger RNA. Messenger RNA is going to be read in sets of 3. We went over that with our activity on muscular dystrophy. And it's going to go from the nucleus to the ribosomes in the cytoplasm. All right, if it's free-floating ribosomes, these proteins can be used in the cell. Ribosomes that are stuck on the ER are going to be make proteins that can be sent out of the cell. What is a ribosome? It's these little dots when you labeled cells, etc. What they really are is they're structures of proteins and ribosomal RNA that are capable of reading mRNA. So that's what they look like. And here would be the mRNA, a lot smaller. It goes through, and as it goes through the ribosome, you make the protein. Here is the protein. The monomers are the amino acids. So how do we get the right amino acid for each codon, right? For the mRNA. Remember that mRNA is a transcription of DNA. It is a copy of the information in the DNA. If you look at the double-stranded DNA and look at the mRNA transcript, you'll see that it's the exact same as one of the strands, except for that the T's are used. Well, we have this special type of RNA called tRNA that's able to carry an amino acid on top of it. Right? And this is bonded by enzymes and put together. The tRNA has a special bottom. And on the bottom, it has what's called an anticodon. That anticodon is going to be the opposite of the mRNA codon. So here it would be, if this is CCG, this would be GGC. Right? And that's how we get the specific amino acid for the mRNA codon. So how does RNA make protein? Do you remember how to read the chart? So DNA, RNA, protein. So here we go. Let's check it out. Oh, I forgot. Here's our terms. Transcription is where we're making RNA. Translation is where we're making proteins from the RNA. right? You should kind of review that worksheet we did in class for how the process works. Here it is in action. So this would be the mRNA, AGU. This is the anticodon. Notice they're in sets of 3. UCA, and that's going to code for this amino acid. Let's see what AGU codes for. AGU codes for serine. So how did I do that? Right? I looked at my codon wheel, first letter, second letter, third letter. Or I could look at my codon chart. If you take AP Bio, this is what they give you on the test. You do not need to memorize this. A, first letter, G, second letter, and then U would be the third letter, and that would code for serine. Notice how on many of these amino acids, the third letter can be different letters. It doesn't have to be, to code for serine, it can either be AGU or AGC. This is what accounts for the fact that there's 20 amino acids, but there's 64 codons. Now, 3 of them are taken up by stop codons. They're just going to cut the protein off and it'll stop making it. But here, this kind of is what leads to redundancy in the genetic code, but it's always specific, right? AAG codes for lysine. AAG will always code for lysine. That's never going to change. So here's the mRNA. Here's the tRNA. This is in the ribosome. This is called a peptide bond when you join 2 amino acids together, and it's going to make a growing protein. This is kind of getting into real molecular stuff here. Why is this important? Well, then I tried to show you a couple of real-life examples in class, right? And we went over sickle cell anemia, we went over muscular dystrophy, and we went over the difference between Becker muscular dystrophy and Duchenne muscular dystrophy. Okay, that was a lot. You should pause and you should go back through your information. You should check out your POGL-like activities that we did in class and some of the models you made. Now we're going to get to mutations. And the word mutation sounds negative to many students, and we tend to think of them as disease-causing mutations. And as a biology teacher, I tend to show a lot of examples of mutations leading to diseases. But you do have to remember that mutations can be good. It can make the protein better, right? And all of life is based upon mutations from universal common ancestors that get passed on and get selected for in evolution. Okay, but let's get back to mutations in DNA. This was a little tough for those of you that were out at CISA, but I enjoyed these couple of lessons. I think you did a really nice job digging into what are mutations, and then you did good work with muscular dystrophy. So see if you can remember the difference between a substitution mutation and a frame shift mutation. I'll give you a second. All right, now that I wet my whistle, this is an example of a substitution mutation that T was changed to a C. So replace one base with another. Over here would be an example of an insertion. This was the original protein sequence, the original mRNA, DNA on top. Here, you insert a letter, so it's going to shift everything down. The way I taught this in class was the sentence, "The fat cat ate the rat." So if we take the fat cat and we change the C to a B, the fat bat ate the rat, that would be a substitution mutation. It changes the meaning of the sentence, right? But we can still read the sentence. If we do an insertion and we insert a letter before cat, so maybe we make it the fat zaka, we insert a Z, so that'd be zaka, and then the next part would be start with T, and it would just be a whole messed up sentence. So check out your notes on that one. But that's called an insertion or a frame shift mutation. Same thing with a deletion. If we delete a letter, everything's going to shift over. I then expanded upon this with muscular dystrophy to show you that if you delete items in a multiple of 3, then it might just delete one amino acid. But that's going to be tricky. I want you to just get the big picture on this. What are the effects of those mutations? So before I show you this slide, why don't you try and pause the video and describe the effects of each of these mutations and the different effects that it could have. Okay, here's our DNA. It's going to be ATG. That's going to code for UAC, which is going to code for tyrosine. Here we have a substitution mutation in that third nucleotide. Remember, this is a triplet. This is a codon. Each of these individual ones are nucleotides, and it becomes ATA. That's going to code for UAU, which still codes for tyrosine. So it didn't change the protein at all. That's called a silent mutation. Here we go from ATG to ATC, and that's going to be a substitution mutation, also known as a point mutation, and it's going to code for UAG. Wowzers, that's going to code for stop. It stops the production of the protein. That's called a nonsense mutation. Here, we're going to change the first letter. So we go from ATG to GTG, and that's going to change it to CAC, which changes it to histidine. This is called a missense mutation. We changed one amino acid. Now you can imagine if we inserted or deleted a letter, it would shift the whole thing over. One interesting point here I brought up in class is why are there multiple stop codons? And for that, you can head to the Google and check out the ambush hypothesis. Okay, what can cause these mutations? They can be spontaneous. They can be some errors during DNA copying. I believe when each of us are born, I showed you that Sean Carroll video, where we're going to have around 60 mutations from our parents. Or they can be induced if you smoke or if you're around UV radiation, etc. Right? So wear your sunscreen, don't smoke, don't vape. It's all bad for you. All right. Last but not least was gene regulation. Now for this, we just dealt with prokaryotes. You read a little bit about eukaryotic gene regulation, and I might show you a TED Ed about it later on. But let's get into the lac operon. Can you take out the model that you cut out and you got to take home? Some of you did leave it in class, but you have a model. I'll post it again in Classroom and see how the lac operon works, right? And use those analogies. I really had you think in class about different analogies for the lac operon and for the trip operon. All right, let's see what you got. The lac operon is an inducible operon. What does it mean to induce somebody to do something? Hopefully I induced you to watch this video and to practice biology. It means that we try and convince you to do something. So here we're going to turn on this operon. An operon is a set of genes that are going to code for something. In this instance, these genes, beta-galactosidase, permease, and transcellulose, code for the intake and digestion of lactose. So this is a bacteria, this is E. coli, and it's going to take the lactose around it and digest it. Up here is the regulator gene. The regulator is always going to be transcribed or read by RNA polymerase and make a repressor. The repressor goes to the operator, which is like an on-off switch, and it prevents RNA polymerase from reading these genes. In other words, it prevents transcription from happening. Why would we not want these genes to be transcribed? Why would we turn off genes? Well, it's the same reason that the lights are turned off above me, right? It's nice and sunny outside, so I don't need the lights. The lights in my classroom are off right now because there's no students there. I'll turn them on tomorrow when you come to class, right? We're trying to save energy. We don't want to turn genes on unless they're needed. The bacteria doesn't want to turn its genes on unless they're needed. If lactose, actually it's allolactose, it's just an isomer of it, comes into the cell and binds to this repressor protein, it will inactivate it. Let me see if I have nope, I don't have the animation. I'm sorry. It wouldn't work on the YouTube one. But go to my slides I posted in Classroom if you like. So let me be clear. The lactose comes into the cell, it binds to this repressor protein, and it's going to make it fall away from the operator. It's going to turn the light switch on. So we turn the light switch on. RNA polymerase can then transcribe these genes. It makes the RNA, which then makes the proteins to digest lactose. Pretty cool. What's even cooler, what happens when we digest all the lactose? When we digest all the lactose, the repressor protein can now go back to the operator and turn it off. So it turns it off, and then the genes can't be transcribed. Right? This gets even more complicated if you want to get into positive gene regulation with the presence of glucose. Would E. coli rather eat glucose or lactose for dinner? Right? Glucose is already in a monomer form, so I'd rather eat glucose. That is a little more complicated, and you can get into it if you'd like. Well, it's something we go over in AP biology as well. Okay, my daughter has woken up. You want to come be part of the video? So I want you to pause the video and try and figure out the trip operon right here while I take care of Ellie really quickly. Ellie, come here, sweetie. Come here, baby. Hurry. You can be part of Daddy's video. Come on. Quick. Zoom. Okay, Ellie says hi. All right, so Ellie's going to take us through the trip operon right here. Just woke up. So the trip operon, we always need tryptophan in our cell, right? It's an amino acid. We can synthesize it. Or the E. coli can synthesize it. So here, it's going to still make the repressor, but RNA polymerase is not going to be in the operator. It's made in an inactive form. RNA polymerase transcribes these genes. It's 5 genes together, and they're able to make the mRNA and make tryptophan. But let's say you eat a meal high in tryptophan. You have excess tryptophan. Well, then it can turn this off for a little while. Let's save some energy. You just brought in extra tryptophan to the cell. So the tryptophan binds to the repressor and then makes it active. That's the difference between the tryptophan operon and the lac operon. This is called a repressible operon. So the tryptophan activates the repressor. It goes here. It blocks the transcription of tryptophan. I asked you to make an analogy in class, and this one was a little trickier. And I think we settled on 2 of them. One is we live in Oman. It's really hot. It is hot today. I can't wait to play soccer and that, right? And so we always run our ACs. But occasionally we'll get a little storm that comes through and we might turn our AC off for a little while to save some energy. Another example would be as teenagers, you're always hungry. And so you're always making that hunger hormone, ghrelin. But when you eat that meal, you're good for about 20 minutes afterwards, so you stop making the hunger hormone. All right, so check it out. Sorry, that was a little disjointed, but Ellie just woke up, and I'm going to need to get her some lunch. Okay, eukaryotes, we do there's a lot of different ways that we can regulate genes, right? Basically, what I tell my AP students is we can do it at every step of the central dogma. So that is not something you need to know for this class. I just wanted to show you this slide in case you're thinking of signing up for AP Bio. So ignore that note to know at the top. I'll take that off. And in biotechnology, it's not on this test, but it's on the exam. I will go over that because I need to take care of my daughter now and make a separate video for that. But we'll get to do a little bit of it in class. It will be fun. I guarantee. Care and have a wonderful rest of the weekend. And go see the play.